over to Vincent, who will talk to us about plasticity in the multidrug ABC transporter BmrA as a base to handle multiple substrates and how substrates can modulate it for a more efficient use of ATP. So I will now stop sharing, Vincent, and you should be able to share. And I'll hand the floor over to you.
There we go.
Well, I should say, for those of you who would like to ask questions, please put them in the question and answer. And we will either Vincent and Barbara will answer these questions directly, or we will read the questions out. Okay.
Okay. Can you see the screen correctly?
You not yet full screen. Sorry. It's my problem. I think this is my problem.
I see full screen.
Yeah,
yes.
Okay, it's perfect.
Okay, all right, I'm going to start then. So thank you very much for this invitation. It's a great pleasure to share with you the work that we've been doing thanks to Instruct using the EM facilities in Grenoble. And so indeed, what I want to talk to you about is this machine here, this ABC transporter multidrug resistance protein that changes conformation binding ATP here. And what I want to go into detail later on is how substrate binding on this site will affect the ATP binding here and how this whole interplay affects this interconnection between these two conformations. So first, let's look into the biology of this problem. So it's drug resistance in bacteria. And I'm sure many of you know this chart that when a drug comes onto the market, then resistance comes very shortly after, if not already present in the field. So it's definitely a major health problem, and it's an ongoing battle, and we definitely need new drugs. So the mechanisms at play to create this drug resistance are multiple. Basically, the whole idea and the concept is similar in all these mechanisms, that the drug needs to reach its target. And so there's many ways to change this phenomenon. For example, we can modify the target, we can inactivate the drug, we can change, protect the target, etc. What I want to focus on today is really the efflux of the drug out of the cell that reduces the drug concentration inside the cell and just protects the cell. Just want to note that these mechanisms can be single, or they can be multiple acting at the same time in the bacteria. And there's definitely the arrow of time of drug resistance where those mechanisms can shift over pathology. But today I'm only going to focus on drug efflux. So we're going to talk about this protein BmrA for Bacillus subtilis multidrug resistance ATP. And in the context is that the competition for this biotope, which is the soil, there's those two bacteria, Streptomyces tendae, that fights subtilis by secreting this antibiotic, cervamycin C. And Bacillus resists by expelling the drug out of the cell using BmrA. So that's the whole idea. And BmrA belongs to the multidrug resistance type of protein because it's able to handle a wide range of substrates. As you can see, a few examples here. They are very different, and so we wonder how a protein can handle so many different substrates. Today, we're going to focus on this one, rhodamine 6G, doxorubicin, and Hoxt, these three drugs. So it belongs to the ABC, BmrA belongs to the ABC transporter family, specifically the type 4 family, which looks like this, and they all share an alternating access mechanism. So the topology is like it has a transmembrane domain, a nucleotide binding domain. BmrA is a homodimer, so the two halves are identical. Binds the substrate here in the substrate binding site, and then binding of ATP on the nucleotide binding domains. Triggers a closure of them and then a reorientation of the conformation toward the outside where substrate can be expelled. And then ATP hydrolysis resets the transporter in inward-facing conformation. So inward-facing, outward-facing. And to study this protein, we're going to use a catalytic inactive mutant that still can bind ATP and change conformation, but cannot hydrolyze it. So we're just going to look at the forward direction of the movement. So we started this, so we've been working on BmrA for quite some time. So we really mastered the purification. We have a beautiful purification. Back in the days, I say that, but we wanted to solve the structure by X-ray crystallography. So we worked a lot on detergents to really tailor the protein. We worked on a special detergent mixture of DDM and cholate in a special ratio 1:1 that yield a smaller detergent belt around the protein that give better diffracting crystals. But this also thermostabilizes the protein, and it also gives the protein a higher ATPase activity for the wild type protein, of course. The wild type protein can transport doxorubicin, as we can see here, but the mutant is inactive. And so we solved the structure by crystallography, and then we quickly, of course, moved on to cryo-EM, and we solved then many structures using cryo-EM. And so that's we started with the structure and the outward-facing conformation. So we solved several structures, and that's a work including many people. And still today, it's the only structure of ABC transporter with a substrate bound in this conformation. So that really gives us details on how the substrate is released. So we did a whole study where we see the substrate in the substrate binding pocket. What we could see, we can identify by comparing to all the outward-facing structures of ABC transporters. We can look at these two transmembrane domains 1 and 2 that really open to more and more open conformations. Our two structures are here. And what we hypothesize a hand fan motion. You can see that this part is really stable in the monomer, but this part is really mobile. Then we confirm that with molecular dynamics simulation on BmrA, that we can see when there's no substrate, it's really mobile, but when the substrate is present, then the structure is more closed and it's not moving as much. And we also did proton deuterium exchange to show that also this group is much more flexible when in absence of drug compared to the presence of drug. So this drug is rhodamine 6G, and we're gonna continue with this drug for a few moments. So that led us to a first mechanism where instead of going through discrete, well-defined states, instead of that, we postulate that this protein just swings back and forth around an occluded conformation and using its plasticity. We hypothesize that the plasticity in an inward-facing conformation is there based on other structure of ABC transporters, and that's what helps it to accommodate different substrates. And then the plasticity shifts to the outward-facing part of the structure, where then the helix is deformed to be able to release the substrate, and then the hydrophobicity of the cavity recloses it to go to the occluded conformation. ATP hydrolysis occurs only after to reset to the inward-facing conformation. So that was the starting hypothesis of what I want to go into detail now. And then we wanted to understand more the plasticity of the inward-facing conformation. So we continue with that same catalytic inactive mutant that's only going this forward direction. And we looked at the binding of ATP, just the binding on the nucleotide binding domain. And since the two halves are identical, we can see that the binding follows a hyperbolic curve, so a Michaelian type of binding with a KD of 150 micromolar, completely fine, and as expected. However, when we incubate with the drug prior to ATP binding, then we see that we shift to a sigmoidal type of curve, so an allostery going on between the substrate binding site and the nucleotide binding domains, which are no longer identical. So it's a homodimer, but they behave as if they are different. So we were curious about that. We wanted to understand. And also, it shows an apparent increase in affinity about twofold for ATP. This is relevant because these two bacteria, as I told you, are soil bacteria, and it's a nutrient-deficient media. And so ATP concentrations in the cell, if you're growing in LB in exponential phase, then you have 5 millimolar ATP in the cell. But if you go to nutrient-deficient places, even in LB in the lag phase or the plateau, then the ATP concentrations drop, and actually this concentration range becomes very relevant. So what we're going to look at, this transition is actually corresponds to to relevant conditions in vivo. And so what we see is that we switch from one conformation to another on a very narrow range of concentration in presence of drug, while it takes much longer to reach this in the absence of the drug. So we did this structural enzymology approach where we said, okay, we're going to decipher this by looking at the different intermediates. So we started collecting points. So those 2 points at saturating concentration, that are the starting point of the study, and that's what we collected previously, the structures I showed you. And then we went down, we collected without drug APO, completely without ATP, then a 1-to-1 molar ratio of drug per dimer, and then at the KD or the K05. And we see that the protein starts at 100% of the particles that are inward-facing conformation. And then we go to 100% outward-facing conformation at saturating ATP. And then there's interesting point that, for example, the 1-to-1 molar ratio in presence of drug here, then we end up with 25% of the population in an outward-facing conformation. That shows that this entity binds to ATP and basically collects all the ATP in the sample and switch to the outward-facing conformation, and it's locked there. In accordance with the allosteric, the fact that when one ATP binds, it helps the other one bind faster. And then when we go to the K05, at 70 micromolar, we have 60% of the particle in the outward-facing conformation, and it requires more ATP to reach this in the absence of drug, which is in accordance with the enzymology. So then we wanted to understand what's going on. So we refined models for each high-resolution structure we had. So I want to start with this one in outward-facing conformation. It's very interesting because here we have 2 structures drug-bound, which are here, and 2 structures without drug. And so it represents a duplicate of what we saw previously, and we really see this hand-fan motion going on. So we have a duplicate of the previous observation, so we're really happy. And it's pretty rare to see that, but now with cryo-EM, we can do multiplicates of structures. So that's quite interesting. And so it backs up our claims that we made originally. In an inward-facing conformation, and we can really see the plasticity going on. So we did this alignment on this side of the protein, and you can see that this side is really well aligned. There's 4 structures, so they are all well aligned. But on this side, you can see it's much more crowded, so it's difficult to see. That shows the movement. You can see the movement from under here, and then we undergo a whole rearrangement on the 2 nucleotide binding domains that become, in the presence of the drug, more facing each other compared to in absence of the drug, where they adopt a little different conformation. Something interesting also is that the drug binding site is here as expected, and it's more or less the same as the outward-facing conformation. And it doesn't really rearrange in the presence of drug. So it's the presence of drug really rearranges the whole arrangement of the of this half, but it doesn't change the drug binding site. Then we wanted to know, okay, so how does the protein move? And I think that's the really interesting point of this whole study. So we did 3D variability analysis of BmrA in those 2 conditions, in the APO form, meaning without ATP, but in the presence of drug or in the absence. First, to be able to compare similar things, we just checked that indeed we have the same particle distribution, and also in latent space, we have also a similar distribution of variance across the different coordinates. So we can really compare those 2 forms. And what we see when we did these 3D variability analysis, for example, 3D VA in CryoSpark, then we end up with these beautiful motions. And we can see the protein move, and it's wonderful. I think it's really amazing, but it's very difficult to analyze. So for that, I created this software called Variability Refinement, VARREF, and it's available in Phoenix. So we teamed up with Pavel Afonin to do that. And it basically refines an ensemble of PDB inside this ensemble of maps, and we end up with this. And then we can use standard PDB tools to analyze the structure. So if I take the structure and I overlay them, and I color them just so it looks nicer, what we can see is that we can see the regions that move and the regions that don't move. For example, this beta sheet here doesn't move. Well, here we have a translation this way, and here we have a translation that way. So what we clearly see is the rotation going on. We can see what residue originate the translation. We can quantify it, so it's the rotation. So it's a 12-degree rotation, and we can just measure everything. So we went on and did this analysis for all the components of this of those two proteins, without drug and with drug. And so if I look at the nucleotide binding domains, what we can see is that we have a first movement where it's a rotation of 12 degrees centered of this beta sheet, and in the presence of drug, we have the same movement going on. We have a second movement where the rotation is occurring starting on this helix on the side, and we have a 16-degree rotation on this side, and in the presence of drug, the rotation is really decreased by more than half. And then we have a third movement, which is the general motion, breathing motion of the nucleotide binding domain going away from each other. And this breathing motion is much more reduced in the presence of the drug. So what we can see during this analysis is that the drug really reduces the conformational space exploration of the nucleotide binding domains, where ATP is going to bind. I just wanted to show, but I'm not going to go into detail, that you can also overlay those different things. Sometimes it's easier to compare because you can use standard PDB tools to really understand your movements. I'm not going to, it's the same things, I just, sometimes it's easier to look at them separately. Then we wanted to validate our findings, so we also did molecular dynamics simulation, completely unrestrained. So what you can already see is that looking at all the frames accumulated is that there's much more movement going on in the absence of drug compared to the presence of drug. It's also shown here using the arrows that just show from the beginning to the end of the simulation for a simplification. But you can see that in the absence of drug, we did 3 replicates of 700 nanoseconds each. You can see that the nucleotide binding domains explore a wide range of conformations, while in the presence of rhodamine, it's much more focused and much more, much less exploring. So that really backs up the claim. So what we see structurally that makes the difference between those things is that the drug really reduces the conformational space exploration. So then we wanted to know, okay, so that's interesting. That's what we see with the structure. And what's the effect on function? So we went back to function. And there's 2 things that are really interesting for these ABC transporters, that we can measure the way they hydrolyze ATP. They have a natural tendency to hydrolyze ATP. And for example, at 1 millimolar, which is corresponding to this part here, we're really at the plateau. We don't see any effect of the drug. We can see that it hydrolyzes ATP the same way. But when we place ourselves at 300 micromolar, so here, then we can see an acceleration of the ATP's activity, meaning that the drug has an effect on how something happens here. This, we can see it in detergents, we can see it in liposomes, and we can see it in native membranes. So there's really something going on. And then we went on to do to measure transport. And for that, we use 2 different drugs because we cannot measure rhodamine transport, but we can measure HypA transport or doxorubicin transport using fluorescence assay. And I must say, this is haute couture of biochemical experiment. It's really beautiful experiments where we measured at the same time drug transport and ATPase activity, and we quantify the drug transport using fluorescence. And I must say also that this experiment, all these experiments need to be done the same day. So this is a monoplicate, a typical monoplicate of the experiment, and it's been repeated 4 times on 4 different batches that allow us to quantify the amount of drug transported per ATP hydrolyzed, and that gives this curve for HypA and this curve for doxorubicin. So I'm just going to show them, on this slide here. And what we see here, it's actually something very important for ABC transporters, that we definitely have a biphasic curve that shows that in this range of ATP concentration, where we also see stimulation of ATPase activity, that's where we have the most transport of hooks. And then when we go to much higher concentration of ATP, where drugs no longer have an effect, then there's more, the efficiency decreases, meaning that there's more ATP hydrolyzed than the substrate being transported. And that's for ABC transporters very important, because it means that those two activities are actually completely uncoupled. It means that ATP gets hydrolyzed whenever ATP comes, and substrates transported whenever there is this cycle going on. But those two things are really completely uncoupled, except in this range here, where the substrate has an effect that helps. With the low ATP concentration, it's basically at the KD, so there's not that much ATP binding, and that helps ATP to bind and to be transported. And what we can see is the difference between those two drugs is most likely linked to the physicochemical characteristic of the drugs, where Hooks is a lot more hydrophobic, so it's probably much more concentrated in the membrane, ready to go into the binding site. So those things led us to postulate this mechanism where we observe this plasticity of this ABC transporter that is able to bind ATP, and in that case, it binds ATP equally well between those two sites, and then it hydrolyzes back ATP, and that's what we call the native ATPase activity. And in the presence of drug, it really decreases the conformational space exploration. And also, one of the conformational space exploration decreased is this two nucleotide binding domains getting close to each other. So it has to go around the drug, and so it forms the first nucleotide binding domain between those two sites, and that's why those sites become different, and we see the allosteric. And that helps the second ATP to bind, and then goes to the outward-facing conformation and undergoes the same cycle. So that's how the drug helps for a more efficient use of ATP in those low ATP concentration. So this is really relevant because, as I was saying, in the soil, it's very deficient in ATP. So this mechanism at play is really going to give a selective advantage for the bacteria. It's going to help. That's conditions where ATP cannot be wasted. It's very precious. So then the drug helps the transporter to use ATP more efficiently to transport the drug and protect the bacteria. So with that, I'm going to stop. I think I'm just on time. I just want to acknowledge those two talented PhD students who did this work, Alexia Gobet, who did all the EM work, and Loïc Massonnier, who teamed up to do also all the biochemistry work on this project. I would like to finish by being a big thanks to Lefteris and Guy Schoen, who really gave us a lot of access to the EM facility, and that was necessary for this work. And I think we don't acknowledge the capacity and to have a great microscope, but it takes a lot of work. And so a big thanks to them. And of course, also to the cryos, most of the data was collected on CM01. And with that, I'm going to stop, and I thank you for your attention.
Thank you very much, Vincent, for this exciting talk. Really integrated structural biology. We already have two questions. One from Bruno Clouds. You can, if you want to read it yourself, or I can read it. I can read it. Are there any specific side chain interactions happening at the tips of the two alpha helices that interact in one of the conformations? These could contribute to stabilizing the particular conformation amongst the other states.
Well, so I guess you're talking about the variability analysis here. I would say I'm not going to go into details of side chain interactions here because we calculated the movements at 4.5 or 6 angstrom resolution because of the resolution of the original data set. So what I'm really looking at is more the movements of the main chain. And yeah, I think you're talking about this interaction between these helices here, if I'm correct. I hope this answers your question, but don't hesitate to ask more.
So Bruno has replied, the region seems to interact according to the densities.
Yeah, that's right. Well, because in one of the maps, Vincent, you showed the density was connecting between the two tips of the helices. That's why I thought they might be
like this one. Yeah. Yeah, I think that's because that's the I think that's an effect of the 3D variability analysis. But so what's important here is that we feed to Phoenix the high resolution PDB file that we refine against the high resolution consensus map, and then we look at the variation using 3DVA. So yeah, I think this is an effect of the. But I mean, this part here, the connection is definitely something that we have seen even for other structures of ABC transporter. We can do cross-linking. There's a cross-linking possible between those two helices, and that doesn't actually prevent the transporter to work. So it's not surprising. And I think it's
it sounds perfectly fun, but I would even say if you see it in the conformational analysis, then it's really present. So I mean, it's a very nice feature. That's why I thought it could be even such that, because it's a prolongation of helices, you know, like in beta addition motifs, you have that it's side on, and here it's tip on the helices. Maybe there's, because I mean, the atomic model might suggest some interactions that you could have a pairwise hydrogen bond or something like this, you know, because it's a symmetry related interaction, I guess.
Yes.
So
yeah. I think there might be interactions indeed, and in the molecular dynamics simulations, we see those two helices definitely interacting with each other. They probably keep the two nucleotide binding domains not too far away from each other, and at the same time, it's transient enough to be able to kiss and run, so that the whole domain can close on each other. So it's not too strong, not too weak. It's the right balance.
Yeah, very interesting. Thanks, Ian.
Okay, and there is a question also from Arnab Chatterjee. Did you split the best 2D classes of samples with different ATP concentration only into two classes to explore two entirely different conformations? Certainly there will be some conformational change in a particular class. So did you split each class again before going for reconstruction? And what references did you use for 3D classification and refinement?
Okay, so we're talking here. We tried really hard to subclassify in many different things, but basically we couldn't go to other classes than what you see here. So here we don't have the amount of particles for each reconstruction. But for example, for this guy here, we can clearly see in the ab initio that we have many classes of the inward-facing conformation. And when we saw that, we were very excited because we thought we could resolve many different conformations. But in the end, we could not go better in resolution for those classes here. We can only refine one to high resolution. So it's more or less the same for all these things. We could not go to too much subclassification, but in a way, I would say that I'm not too interested in too many subclasses. I'm more interested, and in a way, I'm not even interested. It's a bit of a provocation here, but maybe for discussions. I'm not interested into the high resolution model because we know what it looks like. What I'm interested in is how it moves. So in a way, and it's a little provocation, I must say, we could use an AlphaFold model. It would be just as good. But what we want to see is how it moves. And I think going to lower resolution is what I'm aiming at.
Okay, there is one last comment from Bruno, and then I think we should move on to the next speaker. Have you tried multi-body refinement for that, or MSA, without using any references, direct classification, including focused refinement?
So we haven't tried multi-body refinement, but more or less it's going to give the same results as 3DVA, I would guess. And then we have tried many things in refinement here. We've tried many different things, but again, I think I'm more happy with having one model and see how it moves around it compared to many submodels. But that's something that can be discussed.
Okay, well,
I hope it answers the questions.
Thanks again for a beautiful talk and for very detailed answers. I think we should pass to the next speaker, BmrA Zambelli from the University of Bologna. So BmrA, you can now share your screen, hopefully.
Okay, do you see the screen?
Yeah, so BmrA will talk to us about dissecting the structure of the nickel binding site of HypA urine E2 complex required for urease maturation. The floor is yours.
Hi, everybody. Thanks for the invitation to present this data obtained thanks to Instruct. Today I will speak about this work involving the determination of the nickel binding site between two proteins and how using different techniques, complementary, will help to develop, to really understand how this binding site is in reality, at least as we think it is. So urease is an enzyme that is very efficient. It catalyzes the hydrolysis of urea into carbamate and ammonium, and it's quite important for bioinorganic chemistry. Indeed, it was in 1882 that Robert Koch discovered that Mycobacterium tuberculosis is ureolytic and is the causal agent of tuberculosis. In 1926, Sumner proved that urease is an enzyme by crystallizing it, so he proved that enzymes are proteins. In 1975, Zerner established that nickel has a physiological role for life. It's not just a pollutant, as it's required for catalysis of urease. And in 1983, Warren and Marshall discovered that Helicobacter pylori, that is an ureolytic bacterium, is the causal agent for the peptic ulcer. And in 1995, Hausinger crystallized and determined the structure of the first bacterial urease. The structure of urease is known from bacteria, plant, and in all cases, there is a basic structure that is in bacteria is made of a trimer of trimers, like here in the picture, alpha, beta, gamma multiplied by 3. In some bacteria, such as Helicobacter pylori, this basic structure is multiplied for 4 times. So actually the beta gamma are fused in a single beta subunit, and this trimer alpha beta is multiplied by 4, so it's a dodecamer. In plants, the alpha beta gamma subunits are fused in a unique protein that and the protein is an hexamer of this. But in all cases, the active site is contained in the alpha subunit and is made by 2 nickel ions bridged by a carbamylated lysine and by an hydroxide ion. And in the active site are present other 3 molecules that are substituted by the substrate when it binds to the active site, while the hydroxide ion here is the nucleophile for the hydrolysis reaction. So the enzyme is strictly dependent by the presence of nickel ions in its structure, and it's present in many bacteria. Many of them are good for us, like bacteria that are present in our gut, but many other bacteria are dangerous for our health. Some examples are here, Helicobacter pylori, which infects about 30% of the human population, Mycobacterium tuberculosis, as I mentioned before, Yersinia pestis, that is the causal agent of pestis, and even fungi. Cryptococcus neoformans is a fungi that causes meningitis and other brain damage. And many of these bacteria are included in the report, in the list that WHO made a few years ago of human pathogens that are becoming more and more antibiotic resistant. So 10 out of these 12 human pathogens are ureolytic. For example, in the case of Helicobacter pylori, which is a neurolytic bacteria for which urease is really needed for its growth, for this bacterium, the antibiotic intervention fails in about 20% of cases. So the idea is that targeting urease can be a new strategy to develop new antibiotics. And in the case of pylori, for example, you can see that the bacterium is very spread around the world with, in some countries, even more than 70% of people are infected by the bacterium. And in many countries, the range is between 30 and 50%. So to target urease, we can have two approaches. The first one is to target the enzyme, so to directly target the active site or other parts of the enzyme that inactivate the enzyme itself. But the other idea is to target the mechanism that brings nickel into the active site of urease. And this happens with three proteins, ureD, ureF, and ureG, that build up a molecular chaperone that binds to the enzyme, and somehow it prepares the enzyme to incorporate nickel. Then a fourth protein that is called ureE is the metallochaperone of the system, so it comes and brings nickel into the complex. In Helicobacter pylori, we have another nickel enzyme that is nickel iron hydrogenase that also requires other accessory proteins to incorporate nickel into its active site. And one of these proteins, the metallochaperone of this system, is called HypA. And HypA interacts with ureE and helps ureE to perform its activity. Thus, we concentrated to this complex between HypA and ureE, which then funnels nickel into the urease system. So we had the structure of ureE and HypA. ureE is made of a dimer. Each monomer is composed by two domains, an N-terminal and a C-terminal domain. The C-terminal domain contains very conserved histidine that are here at the interface, and also histidines that are conserved in the tails, in the C-terminal tails. These tails are not visible in the crystal structure because they are disordered. But when nickel binds to the protein, we can see that the histidine present in the C-terminal tail close into the nickel binding site. So nickel is bound by these two very conserved histidine at the interface between the two monomers, and two histidines in the C-terminal tails. And using X-ray absorption spectroscopy, we could see that this is the nickel site, and by ITC we titrated nickel over the protein, and we found that the protein binds nickel with a submicron micromolar affinity, and the reaction is mostly enthalpically driven. Then, using NMR, we determined the structure of HypA. HypA is also made by two domains, an N-terminal domain and a C-terminal domain that usually contains zinc, which has a structural role. When I refer about apo-HypA, I refer about this structure that contains zinc, because the terms apo is referred to nickel. Indeed, if we using chemical shift perturbation, we could find that nickel binds in this region, in the N-terminal domain, and it binds with a paramagnetic mode. And using paramagnetic NMR, we could see several signals deriving from nickel binding to the protein. And by selective deuteration of specific residues, we could find, could identify which part of which atoms are really bound to the nickel site. And in particular, we obtained deuterated methionine and histidine proteins by E. coli expression, while for aspartate and glutamate, we needed to use cell-free expression as a new cell-free expression protocol. So these signals A, B, D, and G derived by histidine 2, and the signals C, E, and H belong to glutamate 3. So we could find that this is the nickel binding site in HypA with nickel bound by the N-terminal NH to the backbone, nitrogen backbone of histidine 2 and glutamate 3, and to the side chain of histidine and of the glutamate. We also titrate the nickel over apo-HypA, and again the KD is around micromolar, and in this case the binding is entropically driven. HypA and urease interact also in apo condition. We could see by SEC-MALS here. The complex formed corresponds to a dimer of urease and a monomer of HypA. And also by ITC, we could titrate HypA over urease, and we found that the KD is around 1 micromolar. And again, here the binding is mostly enthalpically driven, but also entropy is favorable. This is again the HSQC of apo-HypA, and this is the chemical shift perturbation that we had with when HypA is bound to urease. So this is the spectrum of HypA bound to urease, and in the absence and in the presence of nickel. And we could see that the chemical shift perturbation in the presence of urease and nickel occurs in the N-terminal domain, so in the nickel binding domain, while the zinc binding domain remains untouched by this interaction. So we wanted to explore what happens when we add nickel to the complex. To do this, we used ITC, and we titrate nickel HypA over urease. And the binding curve that we obtain is not straightforward. So there are multiple events occurring here, as you can see by the multiple curvatures of the curve. And indeed, there is not only one event that occurs here. There is not only the formation of the complex, because we have also the interaction of urease with nickel, the interaction of HypA with nickel, the interaction of the two aproproteins together, and also we include an additional nickel binding that occurs when the complex is already saturated by nickel. So to dissect this binding, we put together all these events in a global fitting experiments, and we could use this model to fit this curve, and we obtain this KD for all these equilibria. And we could immediately find that here the formation of the complex between urease and HypA in the presence of nickel has a very high affinity. The binding is enthalpically driven, but this binding site was not present for the two proteins alone. So it's a really new binding site. This is the distribution of the species over the titration. And so to understand which residues are really involved in this binding, we mutated the histidine residues involved in nickel binding in urease. And again, we did again the titration, and we observed this curve that could be fitted, deleting these equilibria, because urease, in the absence of this histidine, doesn't bind nickel anymore, we know from the single titration, and the two proteins, urease and HypA, don't interact in the apo conditions. Fitting all this data together, we could see that this binding site for the proteins in the presence of nickel decreases its affinity of 2 orders of magnitude. And also mutating the histidine 152, we did a similar thing. In this case, urease binds nickel, but it also forms a tetramer in the presence of nickel. So we must include this equilibrium, and we obtained this data with that also show that nickel binding decreases in affinity of 2 orders of magnitudes. And here the binding is entropically driven. We also had X-ray absorption spectroscopy data that show that nickel binds to the 2 proteins in an octahedral environment with 3 histidine residues and 3 oxygen-nitrogen ligands. And by paramagnetic NMR of the complex, we could see, you see, this is the complex here. These are the single proteins, nickel urea and nickel HypA. And you could see that the histidine 2 signals are maintained in the complex for HypA, and also the signal of histidine 102 in urea is maintained in the complex. So we could derive this model for the nickel binding site in HypA urea complex. The nickel is bound to histidine 2 side chain, glutamate 3 side chain, 2 nitrogen of the backbone of HypA, and the 2 histidine 102 in urea. And from this, we could derive using quantum mechanical calculation the complex of urea and HypA. So HypA interacts with the region of urea, of course, that binds nickel. And this is the active site that is formed. With this, I finished. I thanked the people that worked in this project from a Laboratory of Bioorganic Chemistry group in the Università of Bologna, Stefano Ciurli and Francesco Musiani, and Michael Marone at the University of Massachusetts for the EXAFs, Mario Piccioli in the University of Firenze for the paramagnetic NMR, and Lionel Imbert and Jérôme Bouvier in Grenoble for the cell-free expression, and Instruct-FR2 for giving us the possibility to use the facilities in Grenoble and in Florence. And thank you all for your attention.
Thank you, Barbara, for this wonderful talk. It's very interesting, very exciting stuff. I have a couple of questions.
Yeah.
Questions. While we're waiting for questions to come in the question and answer. So I noticed in your initial introduction of the system that when the disordered C-terminus binds, I believe, nickel, that it becomes structured, but it looked from your structure that the crystal structure only had density for one of the 2 histidines.
Yeah.
Is that true?
Yeah,
and
yeah.
Do you know why that might be? Is there an asymmetry in that?
No, it's. I think it's mostly it becomes a little bit more ordered, but the tags are not remain unstructured actually also in the crystal structure. So the density of the second histidine was not visible here. So here there is, after the histidine, there is still a tag that is going on. So there are still I don't remember exactly how many, but like 10 residues that are not visible at all. And the other one is not visible.
And the disordered C-terminus, so I think you said it was 20 odd amino acids, is that right?
Yeah.
So does that is that completely disordered in solution in the APRO state? I guess you did NMR of this.
The only thing that we think it's disordered because it's invisible in the diffection map in the crystal structure, and this occurs for all urea for which we have the crystal structure, so also for other urea from other sources. And I'm not sure that we run disorder prediction on this. It's possible. But it's
but do you see it by NMR or you don't see it by NMR?
I don't think we had done this.
Okay. I had one more question, which may be a little bit more technical. So you use the labeling procedure, which is very elegant, to identify the amino acids that were involved in the binding. That's as far as I understood. could you also have used saturation transfer? I mean, the old days, when NMR spectroscopy started off in unlabeled samples, but we saw, as you've shown very nicely, these beautifully shifted residues that were in the vicinity of paramagnetic centers. And of course, by saturating those peaks, which you do see, you can then see, doing some kind of saturation transfer experiment, you can then see which residues are in the diamagnetic form, which are exchanging between these. And this can tell you something about the exchange between the two forms. Did you think about doing this? Have you looked? You could maybe look at the kinetics of the exchange and of the binding of the metal.
I don't think we have done this.
Okay, and it's something that maybe could be. Anyway, I see there are other questions in the question and answer. Sonia Longi. Hello, Sonia. Thanks, Barbara. Very interesting. Could you please comment on the reactions? Comment the reactions entropically driven versus those driven by enthalpy. Yeah. So what that was also.
Yeah. One possibility and one thing that we see is that the when in the model, I go here. I go back here. I mean, this is just speculation, so that's not really, I mean, experimentally validated. But what we see in the model is that when HypA interact with urease, there is a little bit of increase of disorder in the N-terminal pair where the nickel is bound. So this change in entropy can be derived by this, partially disorder of the system, and this can, of course, facilitate binding because entropy increases. I mean, is positive for binding. Let's say. I hope I've answered the question.
Yeah. There's a question from an attendee who is anonymous, but who asked the question: in the ITC measurements with mutants, do the KD values correspond to the equivalent complexes in the wild type protein?
Correspond? I'm not sure I understand the question. In the ITC, do the KD values correspond? I'm not sure I understand the question.
Maybe it's a question about the comparison of the KDs measured in the mutants and the wild type. But I'm not sure. Maybe the person.
Yeah. I mean, I try to clarify what we did here. So we measured the binding curve for all the single interactions, so the urea alone, the HypA alone with nickel, and the two proteins together, so the binary, let's say, complexes, and then for the ternary complex involved the three species, and then we put together all the curves in a global fit experiment using affinimeter, and so we could really derive a quite reliable reliable data. Because we put together different experiments to do the fit and to fit them all together, let's say.
Okay.
I'm not sure if this is the question because
it's possibly the questioner can come back to us if not. Let's assume that has answered the question. I had a final question. So you showed, if I understood correctly, I think you were showing the nickel HypA urea-E2 complex and showing NMR of it, and it was quite broad, and there was a number of the peaks, I guess, were weak because of the size of the complex. This one on the right, exactly. Did I understand correctly? So did you think of deuterating that? Would that improve? Because I guess most of these peaks are broad because of the size of the complex. Is that true? Or is that actually?
Let's say that it was enough to do the assignment to find the stuff, the peaks. So probably deuteration would help.
It might improve a lot. Yeah. You might get maybe more information even. Okay.
Yeah. But still, the fact that here the zinc binding side domain is untouched was really clear in both cases.
Okay. Good. Well, then, if there are no more questions, then I'd like to thank both speakers, Vincent and Barbara, for very fascinating, stimulating talks, and everybody for their attention. And I think we're done. Thank you very much for attending.
Thank you.
Thanks. Bye.
Bye.
Bye.